I will move to the next session, which is going to be a discussion on some of the work that I do as a mentor in another program that has some collaboration with Pine Biotech. Am I going to, I need to share my slides right now, right?
Yes, so just briefly, again, thank you for all the panelists. And this is actually going to be the final part of today. And so I do want to mention that tomorrow we will have a chance to hear from some of the next steps and more practical ways of how many of the students that we are interacting with can get involved in meaningful projects using bioinformatics tools, communicating with mentors, finding out interesting projects. And this transition part of today's session is going to kind of build the expectation on how we move from just learning about these exciting fields to practically, what a student or somebody who's beginning to be involved in this domain could actually do practically. So thank you, Gio. We'll have just briefly, I'll just kind of mention what's the plan. We will have Dr. Gepoliano Chaves speak about the SIP program and the mentorship. We'll have Miss Urja Parikh speak about her experience being mentored and participating in astrobiology as well as other types of research. And Mr. Subhrajit Barua will speak about his astrobiology endeavors and what he is up to next. And then finally, I will conclude with some perspective on the OmicsLogic Research Fellowship, on how you can get involved in some of the topics that we discussed today as a contributor, as a mentor, and as someone that can help us build out a pathway for students to get involved in a meaningful way. So please go ahead, Gio. Thank you.
Thank you, Elia. Do you see my screen?
Yes.
Okay, perfect. All right, so because of the work that the science internship program has developed over the years at the University of California, I want to talk about this issue with inequalities. And I generally use it to think about inequalities in the context of my own country, Brazil. I come from Brazil. So in front of you is a picture that I grew up watching. So in the right, we see few people with a lot, and in the left, we see a lot with very little. This is, in Brazil, this is due to historical reasons. And so I think that education represents really a way that we have to counterbalance inequalities that we have in the countries. I was kind of surprised when I realized that there are inequalities in the United States as well, because we all hear of the United States as the #1 economy and things like that. But there have to be politics to address inequality issues in developed countries as well as in developing countries. So I have this question of what are the boundaries between science and education? Because when I was growing up, I really did not see a clear boundary or a clear limit between science and education. So in my mind, up to a point, I did not think that there was a difference between what is science and what is education. So I guess for the sake of simplicity here, I am just assuming that science and education are the same thing. Maybe you do not agree with me, that's okay. But just for the sake of simplicity and the context that I come from, because in my country, in many situations, in many occasions, you can pursue an educational career, but reaching to a faculty position or reaching to a position in academia can be something really, really challenging. And as you may imagine, there can be many mechanisms of privilege in a situation like Brazil. So what I want to say here is that this program, Science Without Borders, is a program that was launched by the government of Brazil about 10 years ago. And the political, social, and economic situation at that moment was completely different from what it is nowadays, from what it is today in Brazil. I'm talking specifically about Brazil. What happens is that there is a need for the countries to invest in sciences and in genomic sciences specifically. So what happened last year was that the government of Brazil has launched this program called Programa Genomas Brasil. And what the goal of this program is to sequence 1,000 Brazilian nationals. Brazil has this particular feature of being the result of the mixing of three main different populations: Europeans, Africans, and Native Americans. So there is an interest in understanding how that has an impact in understanding disease in the country. So what the interest is right now in the country is to stimulate the sequencing of Brazilian nationals. But this brings a problem because sequencing technologies are really, really expensive. And Brazil is not a rich country, it's not a developed country. So sequencing 1,000 Brazilians represents a problem because there is a chance that we are not going to represent the entire population. We are not going to have kids or scientists from a economically disadvantaged background involved in this process. So in my mind, the problem that we have right now in the country is that we need to make science, we need to make education reach many of these different layers of society. I totally agree with Dr. Andino when he says that we need to make science available for all of the population, both young scientists and scientists that are more seniors. But I really think that what we need to do in Brazil, for instance, is a mindset in the educational culture. We need to more and more make science projects available for students in Brazil. And this is with the hope that it will end up helping the public health system to make educated decisions based on the genetic background of the population of Brazil specifically. So this, in my opinion, this overlaps very well with the mission of the University of California, Santa Cruz, because in 2001, the human genome sequence was published in part due to the efforts of UCSC researchers. Here I'm basically talking about Dr. David Hosser, who is considered to have played a very important role in sequencing the human genome. But also the other program that I have also been involved with is the science internship program. I have been served as a mentor in this program for more than 6 years at this point. And it aims to foster the involvement of high school students in science. This program was funded in 2009 by Dr. Raja Garrata-Curta, who is also a professor at UCSC. Both of these initiatives, the genomic research and SIP, grow every year. In fact, we are just having a conversation with the Genomics Institute to design a bioinformatics course for students of underrepresented background. And as part of the effort that I have been trying to do to share the education that I have had access with my national peers in Brazil, I was able to help in hiring PhD students and professors from Brazil to serve as mentors for American students at the science internship program. And this is totally in alignment with the mission of the SIP program, as I will begin to explain to you right now. I consider myself very close to the mission of SIP, and particularly to the ideas that are on the founding, on the thoughts of the founding director, Dr. Raja. And so SIP has this initiative of stimulating critical thinking and research. And the goal is to create equity in what here we are calling STEAM, for science, technology, engineering, arts, and mathematics. And so that part, creating equity in STEAM, is the CREST part. So this is a philosophy, I would say, that was very present at the very start of the program in 2009. Right now, the program has, as I said, it grows every year. It has grown. My understanding from this part, the Starry Night program, is a program where a person that works with astrophysics, Raja is a professor in the astrophysics department at UCSC. What happens here is that researchers that work observing the cosmos, they receive students, not necessarily here high school students, but students younger than high school students. And what they do is that the students talk to the mentor for 1 hour about the research that they are doing. And so what happens here is that the student has an opportunity to stay with a mentor and see how research is conducted. And what the mentor has to do is that he just needs to adapt himself to the idea that he's being followed by a student, and he can just do his research experiment, and at the same time talk to the students. So this exposes students to the idea, to the concept of science, to the concept of getting involved in a science project. And this is a concept that Raja brings to the mentors all the time, that a science project is very important for a student to work with. This has led to further developments in the SIP program. So with these Python and research tutorials called PyR, what Raja does is he teaches students in basically an introduction to Python, and they also look into a research paper. They look into a research paper, and the questions that are being addressed in the research paper are presented to the students in these tutorial sessions. Then I joined SIP, I believe it was 2015. PyR, I actually do not know when PyR started, but just this year we started teaching this other set of tutorials. We are calling them Recombio, for research in computational biology. And the idea here is to basically also teach students coding. I use R and Bash, the terminal, the command line. And here the idea is also present a research project, and then do some coding that answer the questions that are present in the research in the research article. Particularly here, we are during my PhD, I was really interested in variants in the genome in the human genome. But because the SARS-CoV-2 pandemic hit last year, we switched to studying mutations or variants in the genome of SARS-CoV-2 because it is really many times challenging to work in a computer server with human genomic sequence. So because the genome of the virus is smaller than the human genome, I decided to switch to the virus. It's easier to work with viral sequences than human sequences. So I told you initiatives. I presented to you initiatives that try to stimulate a scientific character in students, and I told you that right now we're developing the idea of doing this even before high school, but SIP really works with high school students, and really when the student comes to an internship with a mentor during the summer with SIP, what we do is we have the students follow us in the lab, doing an experiment, and also doing some coding experiments. As I mentioned just last year, we changed a little bit to studying the SARS-CoV genome rather than the human genome. But really here, the idea is to reinforce the need to expose students to the concept of a scientific project so that they decide or they have the experience to develop a scientific project, and they can pursue a career in science, maybe in a university or maybe in another institution that allows them to develop further their scientific career. So after this all ends, there are initiatives that SIP work with. For instance, the Global Sphere Network. SPHERE stands for STEM Programs for High Schoolers Engaging in Research Early. And it is a combination of programs that intend to mentor high school students. And also along the years, other initiatives have joined forces with SIP. So, sorry. What we have right now is the alumni buddies, which is basically more senior high school students that have been mentored years before the current students. They provide some advice on the research presentation, on the many times these students are using the dorms and the buildings at the university. So things like information about places to eat or how to go for the extracurricular activities that are provided by SIP. This information that is provided by the alumni bodies initiative. This SIP is for teachers in SIP, and the idea here is expanding the SIP initiative to SIPs. I believe it's teachers in the county. CSIP is Community College SIP. The idea here is to provide community college students with some exposure to a scientific project, to working with a mentor or working with a professional in academia. So in conclusion, I think I need to finish here. I think that education and science have the potential to help in correcting historical social inequalities in both developed and developing countries. This maintains an ecosystem that is necessary for the social structure in both these types of countries. And the goal here, I believe, is to provide access to education and to medicine to all individuals in these societies. Particularly, I believe in developing countries, the governments need to have initiatives to invest in the formation of human resources to correct for these historical inequalities and also to maintain a minimum access to education and medicine. And I believe this is where I fit because I don't know if I told you, but my studies were funded by the government of Brazil. And so I need to work to allow education to be available for the Brazilian society. So I would like to end here and thank you so much for this opportunity. Oh, I just finished my slides. We see that Pine Biotech also works to connect students to academia and to professionals, to the academia professionals that are designing the curriculum and the opportunities that shape the future of bioinformatics research.
So, Subhrajit, thank you, Gepoliano, for your presentation.
Thank you.
And I do want to mention that Geo is also starting to mentor a couple of students in one of our programs, which I'll talk about in just a few more minutes. But now we want to hear from Subhrajit Barua and kind of hear about the way he got interested and got actively involved in bioinformatics and astrobiology.
Thank you so much, and I'll share my screen real quick. All right. So first of all, I would like to thank the organizing committee of Pine Biotech for providing this opportunity. And as the theme for today was the changing role of bioinformatics, I would like to talk about how bioinformatics is being used all over the globe to leverage astrobiological studies and the researchers of astrobiology all across the globe. So before we move on to the research of going on the projects and what I'm doing, first of all, a brief introduction of who am I and what am I currently doing. So I began my bioinformatics journey from Pine Biotech itself. So I am an ex-research intern. It is here where I got to learn about the various algorithms, got to learn about transcriptomics, genomics, multiomics, data science, machine learning. And this is really the place where I was baptized with bioinformatics, data science, and its biological applications. After that, I moved on for my higher education, and currently I'm a master's student pursuing molecular biology and biotechnology from Itmo University, St. Petersburg, Russia. And I am also a 100% government funded scholar here from the Russian government, which is a big opportunity given to me for studying getting higher education and also doing a lot of research. Talking about research, and I am here enrolled in the SCAMP International Laboratories at University, and I'm a member of the Applied Genomics Lab, and my supervisor is Dr. Alexei Komisarov. And under him, I am working on a couple of projects involving human transcriptomics, epigenetics, and also certain astrobiological experiments and research projects about which I'll be talking in just a while. Apart from that, since I love astrobiology so much, I am a part of an initiative, startup in India, which is called Spaceonova, and I'm currently the head at the Department of Astrobiology. At Spaceonova, we have a vision and a mission to make India one of those spacefaring ecosystem in the world where each and every individual has the capability and the resources to become a space technologist. And since I love to communicate and spread the word of astrobiology everywhere, I'm also an ardent scientific communicator at Art of Inquiry, and I'm working under Julia Brodsky. She is the CEO of Art of Inquiry and also a former NASA astronaut trainer. And here you can see some of the examples where I have been doing this science communication stuff, basically in the domain of astrobiology. So that's like a lot of information about me, but let's focus on what I'm going to talk about today, that is astrobiology. So if I'm asked to define the term astrobiology and what is it actually, well, a very crisp and short answer would be it's a very multidisciplinary domain of science which involves a lot of, you can say, subdomains or parts of science. For example, origin of life on Earth itself, it's still a mystery and it's being explored. How did life originate on Earth? Similarly, we also have this very curious question which we try to find the answer of, that is life there outside anywhere of Earth? Is life there in any of the other planets or satellites or any of those discovered exoplanets? Because just like in today's talk, Dr. Das Sharma pointed out that there are so many other Earth-like planets all across the universe. So who knows, maybe in one of those planets life do exist. And to understand that and to find out if life does exist somewhere else, it is very essential to study the geology and environmental conditions of those planets. So that can be considered as one of the domains of astrobiology. And finally, the domain in which I am most interested and excited about is space health. There's a lot of risks involved with space travel, and with more and more focus being given on long-term missions like a mission to moon or even to Mars, is going to take a lot of time, and exposure to certain factors for such a long time will have an effect on the human health and the human body. So I'm really interested to investigate and to prepare ourselves before we embark on such a mission, so that this kind of mission will have the least impact and the most outcome in a positive manner. So talking about origin of life, well, there has been a lot of talk going on in the recent days that life might be there on Mars, or maybe if not Mars, some of the moons of some of the icy giants like Europa or Titan. But till date, with all the scientific advancements that we have had over the past many years, our dear planet Earth is the only place where we know that life exists and exists in harmony, grows and prospers everywhere. So, talking about the life on Earth, extremophiles, just like Dr. Das Sharma pointed out, are those categories of microbial organism which really poses a question to us: how much do we really understand life? What is the extent of life and what is the range in which life can survive? Well, for a lot of time, I used to believe that life is confined in a very short range. But how wrong was I? Because talking about the extreme condition of a volcanic eruptions, very high temperature, poisonous gases, lava coming out, life should not exist. But we do have species of bacteria like Streptomyces thermophilic, which do survive in such high conditions, high temperature conditions. Talking about a highly acidic or alkaline lake, life should not perform very well at such an absurd pH condition. But again, we talked about certain extremophiles, for example, Acidothiobacillus thiooxidans, which can survive. And Dr. Das Sharma also gave wonderful examples of so many extremophiles with so many unique characteristics and properties. And finally, this gave me a real shock that for many years I used to believe that life can never survive in space, out of Earth's atmosphere, in the vacuum with so much of radiation and fluctuating temperature going on. Life should never have a chance. That's the reason why we wear spacesuits. But this microorganism, Deinococcus radiodurans, survived for almost a year, exposed completely to the harsh environmental conditions of space aboard the International Space Station. This was a part of the Tanpopo mission by JAXA, and this really opened up a new horizon and a new boundary of exploration. And that's how I have been so much excited about astrobiology, because this really begs us to ask the question: how much do we really understand life? What is the extent of life? As I was saying earlier, I'm really interested about space health, about taking care of the health of astronauts, to understand what happens when somebody goes on a space mission. There are various factors which play a role, and one of the most exciting factor is microgravity. because once we go out of Earth's gravity, very strange things begin to happen. These are some pictures which we are all familiar with. We have seen these sort of pictures all across the internet. But if you notice, some strange phenomenon happens once the acceleration due to gravity, which we are all habituated to, that is 9.8 meter per second square, ceases to exist. For example, a droplet of water does not fall down because there's no gravity anymore, but takes this sort of a circular droplet shape because of the now dominating surface tension force. Similarly, there are a lot of fluids in our body, for example, our blood or cerebrospinal fluid. So this is a question which I always ponder about. What happens to those fluids in our body? There's also fluids inside our cells, which play a role in signaling and stuff. So there's a lot of hormones and enzymes. What happens to those chemicals once a person travels to space and lives in the condition of microgravity? So even though space travel seems very, very exciting for all of us, there's a lot of challenges which these astronauts have to face. For example, altered gravity. There's both microgravity as well as hypergravity. Then there's a high dose of ionizing radiation. There's low pressure and low oxygen, which can act as stress factors. The unusual condition of isolation does have an effect on the psychology and the physical nature of the astronauts. And definitely there's a really altered atmospheric gas concentration in space. So there has been a very good amount of questions going on today as well during the symposium, that there's a lot of things going on in the space biology domain, but is the data available for us to analyze? And how can we actually use bioinformatics to leverage those sort of studies? So here's the answer to that, because NASA and other space agencies have been looking at space omics for a long, long time. But it is only recently that the growing interest of astrobiological research among other researchers as well as students have really forced people to form consortiums and groups such as the Gene Labs and International Standard for Space Omics, who really take care of this sort of data and curate them and make them publicly available for research. This typical idea was really fostered by the twin study, a unique opportunity for mankind to study what happens to humans once they are sent to space. So they were the Kelly brothers, and they are identical twins. One of them served as a ground control on Earth, and the other twin went to space and spent some days, less than a year, about the International Space Station. And once he returned, their DNA was taken, sequenced, and the various omics domain were investigated. There was genomic study, transcriptomic study, study of the telomere, and also multi-omic study. And those sort of studies really gave us the idea that there's definitely certain changes that can be noticed in the person who traveled to space. Some of them resolved after a span of 6 months or so, whereas some of them were really permanent. And they were mostly epigenetic studies, changes in the length of telomeres and stuff. So with these sort of really crazy projects happening all across the globe, me as an enthusiast of astrobiologist really got interested in space omics. And currently I'm really in the beginning of looking into this particular domain. And I am investigating the gene expression of various model organisms, plant model organisms like Arabidopsis, and as well as human cell lines with the help of publicly available data. And I'm utilizing bioinformatics and machine learning algorithms like PCA and hierarchical clustering to generate meaningful data to study the differentially expressed genes, to study the pathways which are altered. And with that, I would like to say that that is like the outline of my projects that I'm currently working on. I'm depending upon the publicly available data in the various consortiums and various repositories, and really comparing them with spaceflight as being one of the conditions versus the ground control, and its effect in isolated factors like isolated microgravity or isolated ionizing radiation. There's different organisms that I'm studying, for example, model organisms like zebrafish and mice and Drosophila, Arabidopsis, and also some human cell lines. And there's a need for this field to grow. There's a lot of opportunity, and with the correct mentorship and correct guidance, I believe more and more students all across the globe can really learn about astrobiology and contribute big time to this growing field of science. And me as an enthusiast would definitely love to contribute and do some more research and to understand what actually happens and how we can address some of the problems that originate when long-term spaceflight is taken. So thank you so much for having me here. And if you have any questions or if you have any suggestions, any feedbacks, definitely feel free to reach out to me.
Thank you very much, Subhrajit, and great to see both the presentation that you prepared and great to interact with you about your project. I wonder if you could introduce Urja. I'll just briefly say from my side that, Urja represents one of those people that has a lot of determination, as far as I can tell, to get to where she needs to go. And as a result, was able to apply herself in many different areas. Only some of them include bioinformatics, from which I know, but I'm looking forward to hearing about your talk. Please go ahead.
Yeah, so to introduce Urja, well, she is a visiting scholar at Blue Marble Space Institute of Science and also a research intern at Pi in Biotech. She is really passionate about astrobiology as well, just like myself. And she is doing some really cool projects and trying to explore more. So I think it's better to hear from Urja herself. So I won't spoil anything. It's over to you, Urja.
Thank you so much, Elia, and thank you so much, Subhrajit, for that lovely introduction. I would like to begin with sharing my screen. All right, so Subhrajit has given a really nice presentation on astrobiology and how we can apply omics to astrobiology. And as part of my project with Blue Marble Space Institute of Science, I am working on Dinofocus radiodurans, which is an extremophile, which Subhrajit also mentioned. And I'm working with Dr. Dimitra Atri, and I have collaborated with Pine Biotech to do my project. But before I start talking about it, I would like to first introduce myself. So I recently graduated in biomedical science from Narsee Monjee Institute of Management Studies in Mumbai. And I will now be beginning my PhD at the German Cancer Research Center, which is in Heidelberg, Germany. I'm also, as already mentioned, a visiting research scholar with Blue Marble Space, and I am working on astrobiology projects over there. So I started my studies in biomedical sciences in 2016, where I gained a lot of knowledge in many different areas of biology. In 2018, I worked at a lab in Mumbai, where I was involved in several different projects, where I also discovered new strains of yeast and UV mutagenized them to make them sensitive to a particular antibiotic. And I was also involved in creating a shuttle vector, which was expressing a type of gene from yeast that made it red in color. I then worked in the field of nanotechnology, where I synthesized zinc oxide nanoparticles from a plant extract. The plant was Tinospora cordifolia. In 2020, I collaborated with Pine Biotech as a research fellow. And with that, I had my first omics project ever. And I think that project really boosted my career, where I tried and elucidated certain biomarkers that are highly expressed in glioblastoma stem cells. Next, for my master's thesis, I worked at a hospital, and I'm also currently working there, where I am involved in several studies. One wherein I am trying to establish certain biomarkers for diseases like intracerebral hematoma and traumatic brain injury. And I have also used machine learning to create a diagnostic system that can it can diagnose the rise in serum creatinine in patients. And now I will be starting my journey in at DKFZ, where I'll be pursuing my PhD in cancer biology. So just to give you an overview of the first omics project that I pursued with Pine Biotech, glioblastoma multiforme is a very aggressive type of brain cancer, and a patient has a survival period of only 15 months post-therapy. What I intended to do was to try and identify the origin of this particular type of cancer. And according to current hypothesis, this particular type of cancer can originate from either cancer stem cells, or it can originate from normal neural stem cells that have undergone some sort of transformation into a cancerous phenotype. So in order to solve this question, I performed RNA sequence analysis. So I took a data set from the GEO database where I took 40 samples, 20 of normal neural stem cells and 20 of glioma stem cells, and performed RNA-seq analysis. What I observed was these two types of stem cells were genetically different, and there was a particular outlier sample that had certain similarities between normal neural stem cells and glioma stem cells, which I believe could be an important hint, as it could give an important clue regarding how normal neural stem cells are transformed to glioma stem cells. I was able to obtain 192 genes that were significantly upregulated and 156 that were significantly downregulated in glioma stem cells. From there, I was also able to identify several important pathways that may be driving tumorigenesis in glioblastomas, like the Wnt signaling pathway, TGF-beta pathway, and the VEGF signaling pathway. And apart from that, I was also able to identify several genes that have currently no known association with this particular type of cancer. So I was able to use omics data analysis to identify several types of biomarkers that can be potentially exploited in the future as therapeutic targets. Now coming to my work with astrobiology, with blue marble space. As I mentioned, I'm working with Deinococcus radiodurans, which is an extremophile that I'll be talking about soon. The reason why I have chosen to pursue this particular project and to work with blue marble space is because astrobiology happens to be one of those areas which really interests me, and maybe in the future I would like to even pursue a career in this field. And because the research that is happening in this particular field is not necessarily only related to space biology, but with this kind of research, we can actually help people on Earth as well. And I'll be talking about how that is possible. So the title of my project is Multi-Omics Analysis of Deinococcus radiodurans, and how we can exploit its radioresistant properties for space travel and even cancer therapy. So space is not complete vacuum. I mean, it is, but there are certain other things that may be influencing the health of an astronaut in space. So there are different types of radiations that pose a very massive threat to the health of astronauts when they are in space. And as we progress with advances in technologies that may bring us closer to traveling to Mars, the threats also remain. So we need to evade those sort of environmental stresses that we may find in space. And those are stresses like solar radiation, galactic radiation, and even radiation that may be trapped inside the magnetosphere. Now, the reason why we are trying to evade this particular risk is because ionizing radiation can cause very, very drastic changes in the human body. It can lead to cancer, it can cause loss of vision, it can cause skin burns, it can cause several health problems in astronauts. And if we are talking about long-term space travel, then we really need to find a way to sustain ourselves through chronic exposure to ionizing radiation. So what can we do to protect the body from this particular ionizing radiation? Now, there's several research that is going on where they are trying to focus on radiation mitigation strategies. And what I am trying to focus on is something very unconventional, which is I'm trying to look at Deinococcus radiodurans as a model organism that can survive very high doses of ionizing radiation. So Deinococcus radiodurans is an extremophile that can survive 12,000, greater than 12,000 grays of radiation, which is several times larger than the ionizing radiation that was created during the atomic bomb droppings at Hiroshima and Nagasaki. The reason why this particular species is this interesting is because it has several mechanisms that help repair the DNA once it is exposed to ionizing radiation. And it has several proteins that are involved in DNA repair mechanisms that give this particular organism an edge when it comes to being exposed and survival in conditions with high ionizing radiation. And it also has certain enzymes that give them this particular advantage to survive. But we currently do not know all the mechanisms that are driving radioresistance in Deinococcus radiodurans. So up until now, there have been some studies that are focused on omics data analysis in terms of Deinococcus radiodurans, but we have still not been able to identify very important genes that may be extremely crucial for the process of radioresistance. And the other question that remains is, even if we do find this particular gene that may be driving radioresistance, can it be exploited in humans? So the aim or the rationale of my study is to identify certain conserved genes across radioresistant species of Deinococcus. And I intend to do that with RNA-seq analysis. Even possibly identify gene regulatory pathways via ChIP-seq analysis, which is epigenetic studies where we can see how the genetic environment of the bacteria is modulated in order to drive radioresistance. And the question that I'm trying to solve is, will we be able to identify genes that can possibly not just help us in sort of like using it for radioresistant mechanisms or radiation mitigation in space, but can we also somehow exploit these particular genes for cancer therapy? Because one of the largest threats in cancer therapy is radioresistance post radiation therapy. So is there any way we can exploit this model organism for that purpose? Another very, I would say this could be like a very ambitious thing to look at, but can we then make a synthetic microbiome using these kind of extremophiles and put them in astronauts that would help them survive in space in the long term? So there have been several studies this far, which I'll just explain to you very briefly. So Sai et al. Have tried to expose Deinococcus to very high amounts of radiation, and they saw certain proteins were overexpressed, and they also found certain novel small RNAs that were expressed. What they are saying is that the small RNAs would be important in looking at downstream targets for identifying radioresistance mechanisms. There's another study that has looked at methylation in the DNA of Deinococcus species, where they have looked at how methylation can lead to upregulation and downregulation of several important genes driving radioresistance in Deinococcus. We can actually, instead of just looking at methylation, we can actually do ChIP-seq analysis. And very recently, ChIP-seq analysis has been done for Deinococcus species. There are other mechanisms that have also been looked at with omics data analysis, where they have looked at this particular pathway, which is the MAS-EF pathway, which is another important driver of radioresistance in Deinococcus. However, the treatment that was given in this particular study was with mitomycin and not ionizing radiation. So we currently don't know whether this could be a good mechanism for driving radioresistance or not. So this particular study, which is conducted by Eugenie et al., is also looking at IRRE and DDRO, which are basically transcriptional repressors that are regulating radioresistance in Deinococcus. And this is actually the first ever ChIP-seq study done in Deinococcus, and its preprint is available on bioRxiv. So this is also a very interesting study to look at. And this is the Tanpopo study that Subhrajit was talking about, where Deinococcus species were exposed to the low Earth orbit on the International Space Station, and then they performed omics data analysis with the Deinococcus species to identify certain genes that may be upregulated or downregulated in Deinococcus. Well, so not only Deinococcus radiodurans, but there are other different types of species that are extremely resistant to ionizing radiation that can be looked at. So what I intend to do is comparative transcriptomics, where I want to identify genes that may be conserved across species of Deinococcus, like Deinococcus deserti, Metallilatus, Bobiensis, Geothermalis, the R1 species. So what I intend to do is take the RNA data of all of these particular species and then identify conserved genes, because these particular conserved genes can then be common targets that we can actually look at with confidence and say that they may be important drivers of radioresistance across Deinococcus species. So this is just like, I'm currently working on this project. So it is work in progress. So I intend to complete the study and find something meaningful in the field of astrobiology, and maybe someday it can be exploited for human purposes as well. So yeah, I would just like to conclude my presentation over here, since I think I've also run out of time. But I would say that omics data analysis, as you know, is something that you can apply to many, many vast areas in biology. Not just cancer, not just astrobiology, but actually any possible field that you can think of. So that is something that I really like about this particular area, that once you have picked up this particular skill, you can apply it to any research area of your interest. So yeah, thank you so much. I would love to take questions if you have any, and you can always contact me on my email.
Great, thank you very much, Urja and Subhrajit. It was very nice to hear your presentations, and we hope that we will still see you soon again and hear about the progress. In conclusion of these great presentations, I want to share a little bit more information on how any one of you can actually join the Research Fellowship Program and participate in a research study using computational tools. And one of these opportunities is a research fellowship program run by Pine Biotech. So the OmicsLogic Research Fellowship is a program designed around computational biology for those that are interested not to just learn and acquire skills, but actually develop a research study. And the Research Fellowship Program is designed to provide support and guidance. And what I want to do in today's brief description of this program is explain how does it actually work and how you can get started. So as you heard today, many collaborators from today's speakers were here to offer their perspective on bioinformatics, different areas of biology, and our close collaboration with these organizations allows us to apply kind of a case study design to this program. So I'm really excited about kind of working with a lot of the experts in the field and being able to make this an opportunity that anybody can access. So what in this section, what I want to do is briefly describe what is the Research Fellowship Program, how to join us this fall, how to submit a project on the OmicsLogic portal, and what makes a project stand out. So first of all, where are we in terms of our program? So we are working primarily in the US, but we do have a lot of collaborations outside, including India and Africa, as you have heard in the beginning of today's program. And overall, we've had a sizable experience working with over 15,000 participants across 162 countries. So we really are agnostic about where you live. And obviously that is because we are online. And so you can join this program virtually. The program really consists of tools for big data analysis. So we heard today, Gio was mentioning how they decided to work on viruses instead of, let's say, human data. That is a typical challenge. Many studies, after they exceed a few samples and large genomes, you don't have tools, you run out of space, you run out of RAM. And so we provide that infrastructure to run your analysis on scale. The second component that we provide is mentorship. It's really more than just having somebody to ask a question. It's somebody that can understand what are you trying to do and guide you with, not as a teacher, right? Not as somebody that actually trains you, but as a mentor, somebody that can oversee, make suggestions, point you in the right direction. And finally, we provide training because many people see a gap between where they are and the skills they need to be able to meaningfully analyze data. We provide organized, scheduled training for you to go through independently, asynchronously on your own time, learn about different types of omics data, learn about different approaches, apply different types of algorithms, and therefore kind of develop your skills. And so briefly, let me first talk about some of these resources that we provide as a part of this program and as a part of some of the other programs that we do as well. So one of these resources is the server tBioInfo platform and the OmicsLogic portal. So you probably all have already seen on the symposium page right on top, you have a menu. On that menu, if you click on the logo, you will go to the main OmicsLogic portal. Just by registering there for free, you will have access to a lot of free resources that you can try out and explore some of the other materials that we have. After 24 hours of being on that portal, you will also get demo access to the tBioInfo platform. And as you've seen today, there are many exciting new features that are kind of coming out on this platform. And we also will be showcasing them once they are ready, but already many areas of omics data are available for analysis on that platform. Now, the portal includes a personal profile. And as somebody who wants to do research, this is the place to start. Anybody who is entering this field in bioinformatics has to build up their profile, and they have to do it by showcasing what have you achieved. So if you ask any scientist, what does that result in? Publications, poster presentations, etc. We've modeled our profile around the same idea, allowing you to see how much you have learned, what are the courses and the programs you're involved in, and build out your profile with relevant project presentations. Now, if you don't know anything at all about a particular interest area that you have and would like to learn, we provide scheduled programs guided by mentors where you can learn synchronously by joining live sessions and also utilize online resources. So we have a lot of asynchronous courses starting from the very beginning and ultimately leading you to complete sessions with practice. And so after completing a number of those, you can interact with the mentors and you can actually complete those areas of specialization and earn points. And so that's another metric that we use to communicate with the students and with the mentors that we have, because ultimately we have a process in mind. We want everyone who wants to be mentored, who wants to do independent research, to be ready for that, right? Not to come with nothing to offer, but come with some basic skills that you can showcase. And so you can do that by building up your profile and showing what courses you have completed. Now, we also provide a number of other opportunities that I want to talk about after this. But before I do, I want to ask all of you that had the patience to stay up until now to actually join us and our mentors on our social media platforms. So we have a group on LinkedIn where you will find all of our mentors and all the different connections that you've seen today. We try to invite all of them to join there. If you want to find their information, how to contact them, you will find them in this group. So please join on LinkedIn and on Facebook. We have two places where we have sizable groups of relevant contacts if you want to get involved in these kinds of opportunities. So please join those. So as I mentioned, this is the place where you can build your profile on Learn and then connect with mentors. And through our program, we'll facilitate introductions and make meaningful introductions with mentors if you would like to join. Yes, you're welcome to join the group on LinkedIn. So what is the training part of what we do? First of all, we have some basic coursework that you can learn about bioinformatics and what it includes, genomics, transcriptomics, metagenomics, epigenomics, and data science. How to work with data, how to code in R and Python, how to run big data analysis pipelines, how to do data wrangling and visualization, right? So all of those things are covered in that portion as well. Then we have specialized training, including next generation sequencing, RNA-seq analysis or single cell analysis, and a lot of similar courses that you can either take asynchronously or guided by a person. Finally, we provide introductory projects that you can take to understand a particular domain, like the SARS-CoV-2. I saw a lot of questions today about what will be the impact of a mutation, or how will the mutation affect transmission, or things like that. So we cover a lot of those questions using publicly available data and publications to try to answer those based on the data that is available. Now, as I mentioned, a lot of that is not possible on your own laptop. And so that's why, as a part of this program, and as a part of just registering actually on the portal, you get access to our big data analysis platform. So there you have access to various processing tools, statistical analysis tools, different types of machine learning models, as well as more advanced kind of sections for multi-omics integration and biological interpretation. Now, after this, you're ready for the fellowship. After the basic training, what is essentially the fellowship itself? The fellowship is how to go beyond that. And that's where you get an opportunity to spend 3 months or 6 months to work on an independent project that you design, that you essentially come up with a topic, you then find the data set, perform literature review, and work with a mentor to complete it. What does it mean to complete it? Completing it means that you present that to your peers and experts for feedback. So as you know, we kind of rely on science published in peer-reviewed journals. Well, peer review is an important part that essentially combines working with others and then submitting your findings for critique, questions, and feedback. And so that's essentially the outcome of this program. Now, many people struggle with the first part of that path. And the first part is finding a topic for your project, or choosing a topic for your project. And so what we recommend is kind of thinking about it as the intersection of 3 different areas: a topic that you know and care about, something that will be interesting to an audience, and something that has available data. You can actually find data sets that describe that and provide you with data. So after you do that, you might want to go through some examples, and we provide a number of different practical examples that you can go through, such as epigenetics of asthma, modeling precision treatment of breast cancer, deep searching sequencing of poliovirus, as we heard today from Raul, different types of ecological projects, analysis of the changing immune responses in cancer for immunotherapy, microbiome biomarkers from the American Gut Microbiome Project, whole transcriptome profiling of cancer tumors in mouse PDX models, deregulation of subcellular networks in cancer patients using the Cancer Genome Atlas, different types of biotechnology, agriculture, neuroscience, virology, and finally, your own project. So after surveying some of the projects and kind of understanding how they were organized, now you are ready to start working on your own project. But just choosing a topic like oncology or virology or even extremophiles is not enough. As you have seen in today's presentations, what makes a project stand out, part of it is what are you focusing on? And so an important part of what you can focus on, it comes from literature, comes from the body of research work conducted by others, where you can review available publications and you can understand what they cover and where are those gaps and unanswered questions or controversy or debate where there's disagreement and gaps in current research. And that can help you make your hypothesis more specific and essentially define your unique question. Then we have some resources that we can help you with to try and find data sets to try and address that gap or controversy with a data set. And so, as you know, there's millions of data sets out there that you can survey and identify something that might lead you in the right direction. Obviously, that's not easy. And so that's why we have a Slack channel that we invited all of you to join, where you can start interacting with mentors, following a weekly regular progress report, and essentially have somebody to give you feedback as you develop the project and as you start working on the analysis. Now, again, all of that is following a process that already is implemented on the Learn portal. Once your profile reaches 10,000 points and you complete your profile, then you have to complete a course on how to design and complete a bioinformatics project, and then you will be able to add a project of your own. Okay, and so what are some of the elements of this program? Number one, you do a comprehension and analysis of existing types of data, existing types of literature, and essentially questions that are being asked in the domain that you find interesting. You then learn how to investigate, not based on ideas and just hypotheses that others have proposed, but think critically about the data that you find and learn how to test your hypotheses, explore the data, and find meaningful patterns. And another component is not just doing the analysis, but also being able to describe it. And so that's the final part where you learn to organize your thoughts and ideas into something that is ready to be shown and reviewed by an expert in the field. And so we've defined this as a specific step-by-step process that we go through in weekly meetings and ultimately help you organize all of that information into something that you can put together, assembling a poster presentation or perhaps a preprint or even a publication submitted to a journal. And so as you have seen today, many of the people that spoke are past research fellows like Urja, and you've heard, and she also went through the same process, developing her project, submitting a poster presentation, getting feedback and working through that feedback, and ultimately planning to publish some of her research. In tomorrow's session, you will also hear about some of the other programs that we've conducted where multiple students work together on poster presentations, either together with our team or independently. What does that allow you to do? First of all, it allows you to really practice the skills that you have learned and try to conduct independent research. But also, as you will hear from Urja and many others that you will meet tomorrow, you will see how this helps you stand out and essentially kind of get your attention in the field that you want to be in, whether it's bioinformatics or a particular area of research in a certain community. And so ultimately, many of the people that we have worked with have went on to present, receive prizes, get recognition, and including recognition on our own portal in situations like these, where we bring together others who are interested in this field, experts, students, and others to really provide good feedback and help them kind of understand what they can do more with the type of information and the type of research that they have done. Now, this applies to many different types of students, including high school students, undergraduate students, as well as graduate students. And we've worked with many graduate students, PhD and postdocs, essentially helping them transition from learning bioinformatics to then being able to apply bioinformatics in a meaningful way to kind of work through their research ideas and get meaningful insights, maybe before they generate the data, or maybe knowing what to do with the data they generate. And also, we have worked with a number of students in high schools in the US. In a lot of the programs that you will hear about tomorrow, students have reached out to us and have utilized the resources that we compiled to help them work through a research project. And some of them even completed a full PhD proposal just working on this kind of a research fellowship. So as we conclude, I just briefly want to answer a few common questions that students have when they hear about this opportunity. So can this program be used for the requirement of a university internship? The answer is yes. What is the difference between 3 and 6 months programs? The difference is that if you just want to apply bioinformatics skills to a project, you only need 3 months. If you want to learn about bioinformatics and then do this, you might need a longer time. If you want to complete a full research study, you definitely need at least 6 months. How many hours commitment does the research fellowship require? So the answer to that varies. And the answer varies because some people come from more of a computational background, some come from more of a biology background. After I'm done, you will see some of the posters that people submitted for the poster competition. Many of them came knowing nothing about bioinformatics, but it took them a longer time to get up to speed. Some of them came with advanced knowledge of bioinformatics, but they learned new methods and applied them to an interesting data set. What are the mentorship options? So we do provide several types of mentorship through the program. One option is just meeting in those regular meetings with a trainer and a mentor that can provide you with some general guidelines. Another opportunity is to work with a mentor one-on-one on a project of your own, where a mentor essentially helps you figure out what to do. And the final option is the other way around, is to join a project that is organized by somebody else, which means that an expert says, I want to study this question and I would like you to help. So those are the three options that are available. Can you choose any topic for your analysis? The answer is yes. Can you participate in group projects? Again, the answer is yes. We have many examples for those that work together with their faculty, for example, or with the whole class. And then finally, pricing, payment options, and scholarships. So let me just briefly show you where you can find all of that information. So if you're already signed up on the learnomicslogic.com portal, you can quickly go and explore the courses that I talked about. But right here under programs, you will find some programs that we offer. And if you search here, if you don't find it right here, you can search right here and you will see research fellowship. And under the research fellowship, you will find all of the information that you need, including our upcoming dates that we will post by tomorrow for when this will start, because the current batch is ending on August 30th. So I welcome all of you to explore this and kind of understand how it works. If you have any questions, feel free to reach out to us at marketing@pine.bio.